How do PET scans show lymphoma activity? Imagine a scan that can reveal the hidden activity of lymphoma deep inside your body. That's what positron emission tomography scans, or PET scans, do. They help doctors see where the disease is active and how it's responding to treatment. The process starts with a special radioactive substance called fluorine-18 fluodeoxyglucose, or FDG. This substance acts like a tiny tracer that mimics glucose, the energy source our cells use. When injected into the bloodstream, FDG travels throughout the body, entering cells just like regular sugar. But because lymphoma cells are more aggressive and have higher energy needs, they consume more glucose than normal cells. This means they take up more FDG. Once inside the lymphoma cells, FDG gets trapped because it cannot be processed like normal glucose. It turns into a form called FDG6-phosphate, which stays inside the cell. About an hour after injection, the tracer has accumulated in areas with active lymphoma. The radioactive part of FDG, fluorine-18, emits positrons. When these positrons collide with nearby electrons, they produce pairs of photons, or light particles, that shoot out in opposite directions. The PET scanner detects these photons and uses their signals to create detailed three-dimensional images. These images show exactly where the tracer has gathered, highlighting areas of high metabolic activity. When combined with a computed tomography scan, or CORT, the PET-CT provides both functional and detailed anatomical information. This helps doctors pinpoint the exact location of lymphoma activity and distinguish it from other conditions like inflammation or infection. Because lymphoma cells are highly active metabolically, PET-CT scans are very sensitive. They can detect lymphoma in lymph nodes that look normal on other scans and find disease outside the lymphatic system, like in the bones, liver, or spleen. This makes PET-CT an essential tool for staging the disease accurately, which guides treatment decisions. The scan's ability to detect active disease even before structural changes appear makes it invaluable for monitoring how well treatment is working. If the FDG uptake remains high during or after therapy, it may suggest that some lymphoma cells are still alive. Conversely, decreasing activity indicates the treatment is effective. However, PET scans have limitations. For example, the brain naturally uses a lot of glucose, so it shows high FDG uptake even without disease. This can make it hard to see lymphoma in the brain. Also, certain treatments like steroids can reduce FDG uptake, potentially hiding disease. That's why doctors often use MRI scans for brain involvement instead. In summary, PET scans show lymphoma activity by highlighting areas where cancer cells are burning through energy at a faster rate than normal cells. This metabolic imaging provides a powerful window into the disease's presence and activity, helping doctors plan and adjust treatment with greater precision. If you or someone you know is undergoing lymphoma evaluation, always consult healthcare professionals for personalized advice and interpretation of imaging results.